Today I'm going to try and answer one of the questions I get a lot, which is what does a marine biologist actually do? That is very different, and depending on what fields and specialty you study or you specialize in, you're going to get a really broad range of answers. So starting with what does a biologist do in general, you ask a question about life. So it's usually like a how, a what, a why. So how does this animal move like they do? Why is this animal green? Then you take your question and try and see if anyone's answered it. So you'll do scientific literature searches, you browse different journals, you read books, you search Google Scholar, things like that, and then you see if anyone's answered that question. And if they haven't, you try and answer it, and you try and answer it based off of how people have answered similar questions before. Um, Moving on from that, you'll kind of write out a proposal like, this is why I want to do this thing, answer this question. So you'll say, this is what I see. What I want to know is this, and here's how I think I'm going to get to my answer. So taking that framework, you just kind of go for it, and it never ends, or it never really comes out how you expected. Things always kind of go wrong and change, and it's sort of all over the place. But what different fields will look like, I'll try and answer in a few videos. So probably the big one that most people are familiar with is taxonomy, which is how is life related to each other. And taxonomy has a lot of different branches, like there are different names for the different animals you can study. So I really like fishes. So the name for taxonomy of fishes is ichthyology. So if I study ichthyology, you would call me an ichthyologist. What we look at when we study taxonomy is different traits that are the same and different traits that are different. So if I'm looking at two different seahorse species, I might notice that one of them has a spiky coronet, so like a little spiky thing on the head, and then another one does not, and it's round. And I'm going to go, is this because they're two different animals, or is this because, that individual just didn't have it, just didn't have that spiky coronet? So I will try and look at different traits, like how many spines are in the fin, how big is the eye, how long is the snout, things like that, and see if it falls into different patterns and frameworks that we have assigned to different species. What a taxonomist day can look like is a lot of this. A lot of the time, taxonomy involves having to look at preserved or dead organisms. So these are some different seahorse species. Here's some adults. And then here's like a day 2 or 3 juvenile. All of these ones died naturally. I did not go out and collect them, but a lot of the time with taxonomy, you do go out and you actively go fishing and you will kill the subject animals and then preserve them. So if I were studying taxonomy of these guys, I would put them under a microscope. I would count the number of fin rays. I would measure them. There's a whole list of different measurements I would take. And then I would also take some skin tissue or fin samples, and then we would send that off for DNA work. So, or I might do the DNA work myself. And we'll look at both the morphology, so the physical characteristics of the animal is the morphology. And we would also look at the molecular data for it, which is the DNA. So then we would compare those and see what does this animal match up to and what does this animal match up to, and write out the differences and the similarities. And then maybe if we find something that's different from both of these and doesn't match with any known species, it could be a new species. And I have discovered and published new species before. And normally what you're doing when you find a new species is first you start out kind of by trying to see if it fits into one of the boxes that is ascribed to a species that we already know about. So once you kind of eliminate every possible answer of what is known, then you write about all the things that are different, and then you see if you have enough evidence to prove that it is new. So what my day would look like when I was doing taxonomy work is part of my day would be spent collecting samples. So I'd go out to the beach, I would look through sand and rocks, and I would try and collect the animals that I wanted to study. So I'd collect more of this new species. Then I would take it into the lab. I would look at some of them while they were alive. I would look at some of them under a microscope while they were preserved and dead. Then I would also take what's called biological staining agents. So it's basically dye for skin or fat or protein. But you put this dye on the animals or inject it into the animals, and then they will color different tissues. So it would like turn fat bright yellow, or it would turn muscle tissue pink. And because the animals I was studying were tiny little clear organisms, I needed to do that in order to see how all of the organs attach to each other. So I was looking at a type of flatworm, and it was a lot of my time was just spent like looking at these animals under a microscope and then writing and drawing everything that I found about them. And so I would look at the yolk producing organs and draw out the shape. I would take pictures of them on a microscope slide with a microscope camera, and then I would put them into a computer program where I can measure them. And I would measure like their length, their width, some of the sizes of their organs, and then I took basically as many of these little worms as I could and collected the data and put it all into a data sheet. And then from that data sheet, I created averages. So like this is the average length of this animal, this is the average width, this is the average or the typical shape that I would see of the yolk producing organ, which was a very specific thing to that species. And then from there, I wrote up what I found. And a big part of writing up a new species is you say, here are all the known species of this genus, so this larger group of animals. So from this genus of animals, this is every species that we know of. These are specific traits of all these species. These are the traits of the species I found that are different from all of these other ones. And then I sent it off for publication, and it was eventually accepted after I made some reviews and changes. And so that is what taxonomy is. So your day as a taxonomist, you spend part of your day usually collecting field samples, or someone sends you field samples, and then you spend a lot of your day measuring, counting, looking at things, observing, and sometimes you find something new that maybe we haven't added to descriptions of known species. Maybe someone just didn't notice it, or maybe they didn't think it was important to write it down. But in some of my work, I'll see we have these really good references for known species, but with science you always try and build on what is known and add a little bit more to it. So when I would find a new characteristic or a characteristic that hadn't been mentioned, I'll just try and make the description a little better and a little more detailed of what is known. So you would just write it down and add that to what is known about the animal. And that's basically what a taxonomist would do, or some of the simplified version of it. When you get more into DNA matches and stuff, it does get quite a bit more complex, but it seems like sci-fi. It's actually pretty simple. To do the procedure. Analyzing all of your data and results can get a little bit tricky, but essentially that's what a marine biologist or taxonomist would do, a marine biologist studying taxonomy. And that is it for this